firstly, thank you, Koei, for the nice introduction for me, and thank you, dkNET, for offering me this great opportunity to introduce our recent finding. My today's topic is about leveraging the computational strategy to identify the type 1 diabetic risk and the clinical trial responder status. My presentation today will be consist of two parts. The first part is about our recent finding into the alternate splicing pattern in pro-inflammatory treated cytokine treated human islet and the circulation level into the new-onset type 1D patients. And based on this recent finding into the alternate splicing, we are looking further into the circulation level. So I proposed the second project to dkNET is to investigation the PBMC from Jews with recent type 1 diabetes. And purpose of this project is to identify the biomarkers associated with the risk for type 1D and the clinical trial responder status. So firstly, I would like to go over the first project. And in the beginning, I would like to introduce this fairing system that in this model, we assume the majority of the people who develop type 1D are harboring those genetic risks like the HLA genotype. And it was triggered by the immune activation like the bacterial and the virus, and followed those immune response development of the autoantibody towards the beta cell specific antigen. So in stage 1 is featured by more than 2 autoantibody development, and the glucose level are still normal here. And in the stage 3, what we call is the clinical diagnosis for T1D. It is also the onset of T1D that is featured by the hypoglycemia and more than 2 autoantibody development. So why we are interested in the stage 3 is because a number of the regulatory agents, for example, those drugs listed here has been tested on stage 3. However, most of them have not reliably induced a durable disease remission. Only one drug, tafasitamab, recently showed delayed progression from stage 2 and got approval from FDA. However, the intervention at stage 2 here are still challenging by the case identification. So most new agents are still continuing to be tested first in stage 3 to establish whether there's a benefit before moving to the early stage. So we are very interested in the stage 3, which is the early onset of T1D. We asked the question, is there a role for transcriptomic profile in T1D early onset status? So to answer this question, Dr. Iván Molina and Dr. Mimra, they have utilizing 2 models to answer the question. In a first model, we will treat human islet with the interleukin-1 beta and interferon gamma for 24 hours to mimic the early stage of the disease. And the purpose of this model is to ask the question, what are the molecular pathways responsible for the immune beta cell dysfunction in early stage disease? And can this pathway be further leveraged for therapeutic strategy? And in model 2, we have been looking into the whole blood RNA from new-onset type 1D patients. And in this model, we ask question, what are the transcriptomic signals that released from stressed beta cell into circulation level that can be leveraged for the biomarker identification or identify those diseases at the early stage? And in the first project, we previously finding is focused on the model 1, that we have treated human islet in total of 10 with the interleukin-1 beta and interferon gamma for 24 hours to mimic the early stage. And then we send those samples for RNA sequencing. In this project, we are very focused on the alternate splicing. And we asked question, what are the pathogenic alternate splicing even responsible for the immune and beta cell dysfunction? And by utilizing our RMS to detect those splicing events, and we found in total 5 different categories. And after cytokine treatment, even with a very stringent threshold, we were able to find a wide spread of abundance of the differential splice events. Among them, the top category is coming from the skip exon, and the second change are coming from the mutually exclusive exon. And after that, we have developed our own bioinformatics pipeline to do the prediction into the function of the alternate splicing and do RBP motif prediction. And the third is to do a co-localization analysis between the T1D GWAS loci and the splicing QTL. And fourthly, we have utilizing the algorithm to predict nonsense mediated decay for alternate splicing. And final, after we have those top target, we have been utilizing qPCR and the single molecular fish to validate those top target. So today I would introduce some of the bioinformatics tool we have been utilizing in this paper. Firstly is the RBP motif enrichment analysis. And by this literature reporting that the isolate and the intronic sequencing surrounding those skip exon could modulate the splicing as a silencer or enhancer. So by this algorithm, we are grabbing those sequencing around the skip exon in total of 7 regions and count the chimer occurrence and comparing those chimer occurrence with background to get a Z-score. And then we divide those chimer motif into 2 directions for exon depletion and exon inclusion, which is 2 directions for splicing, and followed by the PCA analysis. So this is the result by this tool, by this analysis that generally this is a PCA analysis result, and those 2 vectors are the vector for the PCA, and each dot here are representative the motif for each RBP, and we are assuming those motifs are more correlated to those 2 directions are responsible for the exon depletion and exon inclusion in splicing. So by this PCA analysis, we found those motifs enriched for the GU motif are responsible for the exon depletion, and for those motifs are enriched for the A motif are more responsible for the exon inclusion. And furthermore, we assume all the motifs that is more than 2.5 standard deviation are significant RBP that are responsible for differential splicing. So we cross-check their differential expression in the expression profile and found 12 RBP that also show statistically significant change in the expression profile. And interestingly, most of them are show increased expression after cytokine treatment, and including those important splicing factor SRF2. And the next algorithm we have been working on is to the functional prediction into the alternate splicing, because we all know that not all the pre-mRNA splicing related transcript will be successfully translating to protein. So it's important to do a functional prediction into the alternate splicing. And in the first part, we prediction into the nonsense mediated decay, which is very important a human surveillance system. Take this cartoon, for example, like for those returning intron category, if the intron is abnormally kept in the transcript, and sometimes it will lead to the reading frame shift, and sometimes it will cause a premature stop codon, which is a PTC here. And if the PTC is further away from the last exon junction complex, which more than 50 nucleotide away, we are assuming it will be a target for mRNA decay mechanism. So we are running the algorithm by utilizing the BAM file and count the intron read with feature and do the intron sequence translation and predict into those NMD targeted alternate splicing by 15 nucleotide rules. And by this prediction, we were able to get 88% of the return intron as a target for the AMD. So it shows the importance for the prediction into the splicing function. And for second part, we have using another in-house tool, Exon Impact. It's a random forest model for integration 31 different protein structure feature into this random forest model. And it could prioritize those tons of the different splicing event to 129 top target with a higher prediction score. And from that, we choose the top target HLA-DMB and seq exon to do the further validation. So just the brief introduction for the experimental validation part, that this exon is the exon 5 of this gene, and it's a MHC class 2 gene that is encoding the lysosomal targeting signal domain. And the splicing of this exon will lead to 4 different isoforms. And we have cross-checked other outside data set to show that consistently increased splicing for this exon, and further validated by qPCR and Western blot show this gene is a co-localization with the LAMP1 in second treated cell alert. So this story just shows us that how our informatic pipeline could be helpful into the prioritizing for those tons of the whole genome and target sequencing, and help you for the next biological underlying mechanism investigation. For another project ongoing right now, as in collaboration with the Raghu and Bobby Joe, that we have been looking into the circulation level. And for the training set, we have 24 subjects. Half of them is new-onset type 1D, another half is a healthy control. And the purpose of this study is to see whether there is a role for the transcription profile, especially alternate splicing in the prediction for T1D risks. So we have been going through very highly depth sequencing to gather sufficient coverage for those alternate splicing. And by bootstrap random forest model, that we further predict it into additional validation set into 24 samples. And here is just the preliminary result for this project, that we have been comparing the alternate splicing RNA, which is in the right-hand side, to the regular mRNA profile into the clustering analysis. So you can see no matter in the whole genome level or in the significant differential express level, that the alternate splicing has shown a much better performance in clustering when comparing with the regular mRNA profiling. And furthermore, we divided those alternate splicing into 5 different categories for the prediction and validation. And from this AUC performance in the validation set, we can see that those top 3 alternate splicing category has shown much better performance when comparing with the bottom 2. So in summary, for the part 1 of our recent finding is that we have shown alternate splicing is an important mechanism for the gene regulation. For the human islet cell response to the cytokine-mediated stress. In the meantime, we show that in the for the alternate splicing for prediction value in the circulation level. And by utilizing those bioinformatics tool, we have been uncovered new role for the alternate splicing in the regulation for the MHC class 2 molecular, especially for the beta cell. We all know that for the MHC class 2, that is a mechanism in the beta cell is not very clear so far. So we are helping to elucidate their underlying molecular mechanism for that. And the bioinformatics tool could be useful for providing the functional annotation, and especially for the machine learning algorithm in the prioritization and do the new prediction for the risk of the T1D. So based on this finding into the transcriptome level, into the pro-inflammatory cytokine treated human islet and also the new-onset type 1D, we propose further to look into the circulation level, especially for the peripheral blood mononuclear cells collected from the children with recent onset T1D. And because of the heterogeneity issue of that, we were proposing to use single cell and sequencing technique and deconvolution method simultaneously to identify those biomarkers associated with the T1D risk and for the clinical trial responder status. So firstly, to mention here is for the heterogeneity issue among those tissue, like this figure to show that for those dangerous cells, if they are treated with the LPS, that those cells within this cell type will undergoing different transition or development status. So heterogeneity has been recently paid more attention, and especially for the blood tissue, that it is almost among one of the most heterogeneous issue among human organ donors or human organs. And take for the peripheral blood mononuclear cells, for example, even it is only 1% of the blood volume, it is considered of the 20 or 30 more cell type, including CD4 T cell, CD8 T cell, and the B cell, and the NK cell monocytes, and the dendritic cell, macrophage, and some very rare proportion of the hematopoietic stem cell. So existing the study looking into the blood RNA sequencing have been utilizing the bulk RNA sequencing. So we all know that bulk RNA sequencing is like a smoothie. You take average of those expression profile from each cell. However, if the gene expression profile is, for example, like this, like a bimodal distribution, and you take an average of them, you will miss the signal here, and sometimes you will also miss those very rare events there. So currently, there has been a lot of single cell profiling technology applying into the immunology part. For example, like the flow cytometry and the mass cytometry. And all days, they have been very successful in identifying those major immune cell type in single cell level. However, they are limited by the pre-select marker and the valence of the primary sequence, and also the surface protein markers. By utilizing the single cell RNA sequencing technology that including those older time SmartSeq and into the civil and Fluidan platform, and into the CellSeq2 platform and DropSeq platform. This technology has been offered an opportunity for us to looking at those cells into a higher sensitivity. However, we all know that a single cell is still very expensive. And so we are proposing to do the deconvolution strategy in the meanwhile. Advantage of that is it will reduce the cost, especially when you want to use it in a clinical trial, which is a large sample cohorts. It will reduce the cost. And for the traditional approach, they are relying on the combination of the pre-select markers, so multiplex is limited. Also, for those clinical sample like the FFP sample, they may not be able easily to dissociate into the single cell suspensions. So a deconvolution strategy just offer a way that you can infer cell type specific change from the bulk RNA sequencing. So in the old way, people are utilizing the gene set enrichment test, which is when you get the pre-select markers from your specific cell type, for example, T cell, then you can rank those signature marker in your bulk RNA sequencing profile. And if the marker are very highly enriched, then you would say that those bulk RNA sequencing enriched for a particular gene module. And nowadays that people have been utilizing the deconvolution method, and like this cartoon is showing that the left-hand side is the matrix from the bulk RNA sequencing, and each row is one gene, and each column is one sample. So here is the matrix you got from your bulk RNA sequencing. And generally, we assuming the mixture matrix are linear combination of the signature matrix as here, which is each row is one gene and each column is one cell, and here contain the average gene expression profile from your signature matrix. And when you got those two, you can deconvolute those cell proportion matrix out in a mathematical way. So generally that deconvolution can be divided into two parts. One is reference-free, another is reference-based. And for those reference, you can either derive them from your single cell matrix, or you can obtain them from your cell flow cytometry cell type markers. And they will provide you cell proportions, and some tool will provide you the expression profile in step 2. This table just summarizes nowadays some of the developed deconvolution tools, including different statistical approach. Most of them are utilizing the least square algorithm, and some of them is requiring to profile the reference profile, and some of them that is reference-free, like the ICTD method. So take the CIBERSORTx, for example, that it has been show very good performance in the immune cell, and in their formula, they are assuming the signature matrix multiplied by the fraction matrix will be approximately equal to the background sequencing matrix. So when you get your marker matrix here utilizing the supported vector regression method, you can get those fraction matrices out. And in the step 2, they are expanding to more gene, not only limited to the marker gene that in this matrix, and you have your fraction matrix at hand. And in step 2, you can get those group model expression profile, and what they call it is a GP profile by this least square regression model by requiring the constraint to be non-negative. So generally speaking, that deconvolution method is to deconvolute those cellular composition from RNA sample by gene expression profile. And in nowadays, there has been some paper talking about when you do differential expression analysis, we also need to correct for those cell proportion change difference. And by some benchmark paper to comparing their performance that least square method and the supported vector regression method and those robust regression approach have a better performance. And for those reference-free method, like the non-negative matrix factorization method, it's an unsupervised approach. But when you incorporate some previous knowledge from cell-specific marker, it would be improve the performance dramatically. So our workflow is that we have been collecting those T1D samples with recent onset from the Raleigh Hospital from high school medicine, and the mean age is around 11, and we compare to another healthy donor for age gender match. And we isolated those PBMC to do the single-cell RNA sequencing. To highlight that is we had collect those blood sample very freshly after 8 to 10 hours fast and within 48 hours of the diagnosis. And PBMC was immediately isolated for the library construction to avoid the frozen procedure that influence on the cell proportion. So the aim one of the study is to determine the immune cell landscape in human PBMC cell and to identify the cell-specific differential expression between T1D and healthy. And further on that, we will derive our signature based on those single-cell RNA sequencing to integrate to do the deconvolution, to integrate with the public data set to deconvolute the cell proportion and also the cell-specific gene expression out. In N2, that is more focused on the clinical trial data. We want to characterize those cell type proportion change with the risk of the type 1D and also the response to the drug treatment over time. Look at those single cell results when we do integration analysis by the CCA algorithm to harmonize those difference between healthy and the T1D. We can get around 100K high quality single cell matrix. And furthermore, by cluster analysis, we found 80 major cell type and including additional 13 subcluster. For example, that CD4 T cell here is considered one central memory cell and 3 subcluster from naive cell, and the CD8 have 1 naive cell and 2 subcluster from effector memory cell, and for B cell we have 1 from memory cell and 3 subcluster for B naive cell. And firstly, we have checked the cell type proportion change between the healthy state and the T1D onset. Firstly, we can see that a T cell will detect with very high frequency, around 20 to 30% among the blood. And when you do the cell proportion change, you can find the proportion of the CD4 TCM cell and was increased in the T1D group, and proportion of the plasma cytoid dendritic cell and hematopoietic stem progenitor cell and platelet cell was reduced in the T1D group when compared to the healthy status. And then we cross-check those clinical laboratory testing from a CBC test. We can see that in the lymphocytes, monocyte, and platelet, they all very good correlate from the single cell and the CBC test result to show that our single cell result is very reliable. And thirdly is that we have done the differential expression comparison between T1D and healthy. This is the heat map. The darker of the color here means the more differential expressed gene here in this cell type. So by comparing those different cell types, we can find that NK cell has a stronger immune response by differential expression pattern. And even when we do the subcluster analysis, that NK cell, two cluster and also the NK proliferating cell has the largest number of the differential expression gene number. And furthermore, do a GSEA pathway enrichment, we found those genes that are from the pathway of the response to virus and response to cytokine has been activated in the T1D onset status. So NK cell in T1D from our data set show begin as an early stage. And from the literature have shown that NK cell has a complex relationship with the autoimmunity and might work as an effector to the regulatory of the pathology. And explanation for this might be the virus trigger resulting in the altered NK cell gene expression. So this result was similar with one of the recent TIDE study finding. We all know that a TIDE study, they are collecting those high-risk infant sample from their birth to tracing them from birth to the age of 15. So they can collect their sample every 3 to 6 months. So by their cell-specific gene expression analysis, they found that NK cell is very strongly associated with the occurrence of the IAA autoantibody signature, and the most correlation coming from NK cell and followed by the CD4 memory cell. So it's very consistent with our finding here. And lastly, just for the heterogeneity that observed in this single cell sequencing, take for the CD4 T cell as an example, that we can find 3 subcluster here. And when you check the subproportion, they are very different trend for the naive cell and the effector memory cell. And when you see the heat map here, that each row is one gene, each column is one cell, that you will find those gene expression difference between naive cell and effector memory cell. And even within those 2 clusters, you further do a differential expression analysis. You found those cytotoxic gene like GZMA, GZMB, and CS3, CR1, those genes has been shown high expression in one subcluster when compared to another. So just show us that the heterogeneity issue is really existing in those peripheral cells. And for our second end of the study, that we will do the deconvolution and correlate those cell type proportion with the change of the T1D status and also the drug treatment response. So just to show some preliminary research progress on this part, right now we are processing those public data within 2 tools. One is CIBERSORTx, which has shown a good performance in the immune cell, and also do another in-house tool, ICTD, because it's a reference-free method. So we want to run them simultaneously to do the deconvolution and come to your intersection part. And for those clinical data that we will utilize in the longitudinal study by the mixed effect model. And we are planning to do the subgroup analysis by the multiple regression model. And we will utilizing the signature derived from our own single cell sequencing to do the deconvolution. So just the first example is that we have been deconvoluted into those PBMC cells from one very old microarray data that they are collecting those PBMC samples from the healthy control T1D samples and the T2D samples. The mean age of them is around 11, so it's very similar with our single cell RNA matrix. distribution. And we having derived our own signature from this data sets. So this is the heat map to show the signature derived from our own. So we can see that those signature can be very different within different cell types. And meanwhile, we have been comparing with another public signature matrix, AM22, to run them simultaneously. And we have running two tools, ICTD and CIBERSORTx together. So we are utilizing the ICTD, the by cross validation tester algorithm, to derive those signature. And generally that we will detect first the rank 1 module and then consolidate it with the co-marker database and come out with the final data set specific markers. We have been utilizing those e-score formula to performance those deconvolution. And generally it is to evaluate how much of the gene expression is explained by the cell proportion change. So by this e-score that we can see, here's some example of our markers derived so far, that for NK cell and CD8 T cell and dendritic cell. And each row here is one gene, and here just show the e-score of all those gene performance by the signature matrix. And here is the cell type average performance that for each cell type, we take average of them e-score. So we can find that across those different cell type, they got a good e-score for around 80% of the variance can be explained by the cell proportion change. And here is the sample level performance that we check healthy T1D and T2D status to see their Pearson correlation. And we found a very good correlation in this deconvolution process, no matter their healthy status or their T1D or T2D status. So furthermore, that I check those comparison between the performance of the two signature. For example, I comparing the deconvolution from our signature comparing to another public AM22 signature. We found a very good correlation among those major cell types. And furthermore, we are utilizing our own signature to run it through those deconvolution method to compare the CIBERSORTx result with the ICTD. So from those two tools, one is a reference-based, another is a reference-free, that we can see a very strong correlation with the performance even into those cell subcluster, including those CD4 TCM and CD8 TM cell. And further, I compare those proportion profile between different disease status. So A-axis here is the healthy and T1D onset and T1D 1 month and T1D 4 month duration. So Y-axis here is the cell type proportion change. And from this panel, we can check those cell type proportion change intuitively for all those different cell type at one time. And by a pairwise comparison, we can see some cell proportion change among the disease status development. So when comparison for the T1D onset with the healthy control, we observed a similar trend with our study, like the reduced PDC cell and reduced NK cell proportion, and increased the CD8 positive TM cell and Treg cell. And by cross-check with our single cell result with this public data, so we could find a reduced proportion of the plasmacytoid dendritic cell in T1D. For example, in our single cell, we can observe a reduced trend here, and also there is an enhanced MHC class 2 activity observed in this cell type. For example, by this Walker plot, we can see those MHC class 2 gene has shown a higher forward change when compared to the background gene. And by the public data deconvolution, we can see the similar trend for comparison between healthy T1D and T2D. And also when you're tracing those T1D onset development status, that there is also consistent reduced proportion change for this dendritic cell. As it is an innate immune system, so And there has been a lot of research into this cell type. So it's a function by triggering those anti-islet autoimmunity in the context of the autoimmune prone environment. And another cell is for the B cell that we have showed the B90 cell has been consistently increased among our data set, and also by comparing with those public data set, we can increase the statistical power by increase the sample size. So we can see there is a very dramatically statistically significant change for the B9 cell during the T1D early onset. And we all know that there has been one drug that anti-CD22 monoclonal antibody rituximab to function as a depleted B cell in the patient with the recent onset of the T1D, and they have been functioning as the preserved beta cell function and delay the requirement for insulin for 1 year of the treatment. So in our next example I want to show here is that we, for our deconvolution research into the this drug for the rituximab into a clinical trial data. And this is the randomized double-blind clinical trial data. And there has been 87 participants from recent onset T1D in this clinical trial, and the infusion of the rituximab or placebo is on days 1, 8, 15, and 22. So study endpoint is the geometric mean area of the AUC of the scRNA-seq peptide. By this figure, you can see that by this clinical trial, in the 12 months ending for the clinical trial, that C-peptide AUC was significantly higher in the rituximab group, and for the A1C dose was significantly lower from the rituximab when compared to the control arm. So there has been another study from Peter Lindsay group. They found that there is an elevated T cell transcription level could be associated with the poor clinical response following the drug treatment. Based on this analysis, that TriNet has initiated a trial to follow by the treatment of the rituximab for another drug is a T-regulated drug CTLA-4 to treat those patients at the same time. So we are also retrieving those public data from GEO database for their expression profile. And by this, we can get 37 patients from the treatment on, another 17 patients from the placebo on. And also we do those deconvolution by those two tools simultaneously using different signature. We can also find a strong correlation between those two deconvolution methods for those major cell type. And then we look at those cell type proportion change profile and change over time. So here each axis is the x-axis is the visit time from those patients from 26 weeks, 52 weeks, and until the 104 weeks. And the y-axis is their cell type proportion change across time. So by this panel, we can see those cell proportion change for all those cells at one time. And the infusion is at a very early times of that. And the clinical trial date ending point is for the 52 weeks. So we are utilizing the responder here to distinguish those cell proportion change between responder and non-responder. So in this study, they are defined a responder as the change of the AUC of the C-peptide response from baseline to 6 months. And by comparison, those cell proportion change between responder and the non-responder, we firstly found that B cell subset for the B memory cell here was reduced after the drug treatment. And when we do a t-test to calculate the baseline difference, we found that non-responder has a higher proportion of the CD4 positive TCM cell and DNT cell, and the responder had a higher neutrophil cells. And furthermore, I do a linear mix effect model to see those differences. We also find that for those non-responder patient has a high proportion of the CD4 TCM cell and the anti-cell Treg cell, and the responder has a high proportion of the neutrophil cells. So for the non-responder has a higher T cell is also consistent with what Peter Lindsay group previously observation for that. And by deconvolution, we can go to high resolution to look at those cell proportion change across time by a time series analysis. And summary for part 2 is that we have been utilizing those single cell sequencing technique into the PBMC cell, and we were able to identify 31 different cell subcluster, and heterogeneity was a result within each cell type. So by comparison between the T1D and the healthy status, we do observe some cell proportion change. And NK cell, dangerous cell, they have shown early immune response in the T1D onset by those differential expression analysis. So by the preliminary analysis into the deconvolution method, by utilizing our own signature to deconvolute into those public data set, we found a strong correlation between the method CIBERSORT and another reference-free method, ICTD, for very good correlation between them for the cell proportion estimate. And by utilizing this method into those clinical trial data, we show more information when compared to the existing report. And we found that the B cell and the CD4 T cell, Treg cell, and the neutrophil cell was associated with the response of this drug. And this approach has shown us to do a secondary analysis for those clinical trial data. So we are planning to apply this approach into more clinical trial data to link the cell type specific immune signature with the drug responder status. So in our future direction that we will refine the single cell analysis, especially for those subcluster. And we have downloaded some flow cytometry assay data to further validate those findings. And for the deconvolution part, we will do more work into the optimization for the pipeline and with the investigation into additional clinical trial data sets. So we are more interested in further is to determine the longitudinal trajectory to tracing how the C-peptide AUC change with the cell proportion over time. Lastly, I would like to thank you, Dr. Ivans Molina, for her supervision to me, and also she has been helped me for the collecting those clinical samples from the Riley Hospital for our single cell project. And also thank you, Dr. Yunlong Liu and Dr. Mirmar for their previous mentor to me. And thank you everyone who has been contributing to those projects, including Chuanpeng, Shi Zhang, Tim Boguo, and Jinliu Faru, Chijun. They are working on the experimental part for the validation of the splicing events. And also our collaborator, Bobby Joe from PNL. Lastly, also importantly, thank you, dkNET, for offering me this funding support. It is my first federal fund support, so thank you so much. And also thank you, the human donor and their family for their contribution to our research community. And thank you, every member from Kamana Lab. And that's the end of this talk, and thank you for your attendance. I'm happy to take any questions.
Okay, thank you, Wenting, for a great presentation. So now is our Q&A session. For the audience, please feel free to unmute yourself to join the discussion, or you can continue to use the chat to ask questions. And then, yeah. So Wenting, during your presentation, I think there's one question from the chat. Let me take a look. I'm interested in your last deconvolution analysis into clinical trial of rituximab. You have introduced the percentage difference between responder and non-responder. How about the difference between two arms? I mean, comparison between rituximab and placebo arm?
Yes, that we have, due to time limit, that we we have been comparison those proportion change between the treatment arm and the placebo arm that. So by a linear mixed model that we have found those cell proportion, most significant changes coming from the B cell, as the drug here for the rituximab is responsible for the B cell depletion. So for other cell proportion that they are not so much dramatically changed by the statistical comparison. Yeah, for the comparison between the treatment arm and the placebo arm.
Does that answer the question?
Mm-hmm. Yeah, and that's the answer for this question.
Okay, yeah. Okay, so the next question from the audience is, have you tried other deconvolution algorithm? How they performed? What's your next plan for deconvolution analysis?
And yes, we have tried other tools like the API and the music. And by the comparison, we found that CIBERSORT may be the best in the performance for immune cell. And also we want to include another reference-free method like the ICTD to run them simultaneously to because the reference-free method will be more tolerant into those sample differential distribution issue for the deconvolution method. So we firstly choosing those two for the ICTD and CIBERSORT to run in the first run, and by some preliminary analysis into those we have do one deconvolution analysis by utilizing the IPIC and the CIBERSORT to run into those one of our preliminary biomarker sequence data from blood. So from this deconvolution, we can see those IPIC and the CIBERSORT, they are very good correlated with each other. But for IPIC, they are only limited to those 5 major immune cell type, and the CIBERSORT is more aggressively into more cell subcluster, so that we have been choosing CIBERSORT firstly to run those deconvolution. And for music, we also try to deconvolution into some clinical trial data, and looks like they are more good at some solid tissue, and for the blood tissue that we found CIBERSORT is better performance. Yeah, this is the end of this question answer.
Thank you. Let me see. So here's another question from the audience. I'm interested in your first part into alternative splicing. What's the difference between those 5 categories of splicing event? Are there any functional relevant enriched pathways difference for those differential splice event?
And for this part, and due to the time limitation, I haven't been introduced those 5 different splicing categories. So generally, they are characterized by their splicing mechanism difference. For example, the skip exon is the most straightforward one that if you skip out, then there will be those 3 exon combination in the transcript. And if it's not spliced out, is kept in the transcript, then will be those 4 exon together to form the transcript. Also for the mutual exclusive exon is that there are 2 exon you can choose. So there are different way of the combination of them into the final transcript. Like this is the first exon and the third exon and the fourth to be combination, and in the top one there is a 1 to 4 exon to be combination into the final transcript. So so basically those are alternate splicing and category are defined by how they splicing, and so they can divide into those 5 different category for that. And And for the functional relevant with that, we have do some differential expression pathway relative with the pathway analysis that I haven't shown today in the talk. I can find some result. There is by the so it's our previous finding. Or it's not here. That for some pathway analysis I've done previously. So it's too much slides and oh, here. Is the function pathway analysis that we have done when combining all those different splicing events by whether it's exon inclusion and exon depletion is two direction for the splicing. So you can see the relative GOBP turn enriched for those two directions for the exon inclusion. It's more enriched for the exotosis and the Golgi-mediated transporter or those phosphorylation function. And for the exon depletion, is more enriched for those basic biological function like the protein transporter, protein phosphorylation, and the cell cycle related pathway and the cell division pathway. So they are from the exon inclusion and the depletion, they will be correlated with different biological process by the GOBP turn enrichment analysis.
Okay,
yeah, that's the answer for this question.
Thank you. And I think that there is one more question from audience. You have mentioned splicing factor by your prediction. Have you done any follow-up assay for that or any plan for this analysis further?
Yes, this is an interesting question for the RBP analysis. That's from this assay, we were able to narrow down to 12 different splicing factor, and especially for those SF2, that is an important splicing factor has been shown, and their role in cancer immunotherapy, cancer therapy. So we have do some the prediction for those splicing factor to see what's those splicing factor target skip exon events by our detection. And for those experiment part, we have do one cross check with one public data. In that public data, we also found that the after cytokine treatment, they are very consistent with our finding that this splicing factor also has been increased after cytokine treatment. And for those prediction and also those public data validation, maybe you can refer to the paper in diabetes that we have more detailed information in the paper for this part. And in the future, that I also think the splicing factor is a very promising field for us to investigate more, like for those CLIP-seq assay or other assay to cross-check those splicing factor performance after cytokine treatment, combined with our alternate splicing finding.